All right, chapter 7, urinary system. Really short chapter. Why even bother? Well, I want to cover every system and the kidneys and how they really function inside, and they're the star of the show here. It was just so complicated, I didn't want to try to get into all the details. But it really, main purpose of this is to review all the different types of membrane transport we've talked about to date. Got a couple more coming up in the future, endocytosis and exocytosis, um, but they're completely different. So I'd like to review this one. But first, let's take a look at the concept map just to talk generally about what the kidneys do. For chapter 7, we just have a little bit of physiology to cover, and some of it relates right back to the heart, which creates this high pressure of blood that flows down into the body. And the kidney's job is to clean up that blood. And so it's come back into the heart, gone to the lungs and picked up oxygen, and now it's ready to return. But when it comes back, it carries a lot of waste with it. Now, some of that, the CO2, is exhaled from the lungs and we get rid of it there, but the rest is cleaned up by the kidneys. So as we travel down here, this blood is still fairly high pressure and flows through the kidneys. Not like this, there's not a tunnel through it, but the kidneys squeeze that blood into these little nephrons, and in there, what happens is some of the blood, all the plasma, the liquid part, is squeezed out and then run through a series of tubes. And what happens in the kidneys is you have primary active transport, secondary active transport, osmosis, and facilitated diffusion, all happening to clean that blood up. And the main thing that we're trying to clean up is urea. Urea goes into the kidneys, and most of it leaves and goes into the bladder, along with a little bit of water. But hopefully, the good stuff that goes in, like water, also comes out the other side. And sodium goes in, sodium comes out. Glucose goes in, and glucose comes out. If that didn't happen, if everything, all this stuff left and went into the bladder, well, we'd drain our glucose supply, we'd feel really low in blood pressure because the sodium would leave, we'd have all kinds of problems. Kidneys work really hard and use a lot of energy because there's so much active transport going on. So it's not a muscle, it's not moving around, but there's still a lot happening at the cellular level. But it's an amazing filtration system. It lasts your entire life, and you don't have to replace the filter. What a great system. Okay, so when it comes to membrane transport, stuff moving just across the membranes, we can divide it generally into two categories, the passive and the active. There are three types of passive membrane transport we've talked about, and two types of active. How do we differentiate which is which? Well, it's passive if things are moving down their gradient. Remember that idea of going from high to low? We even talked about this with flow down gradients. So if it's going downhill, high to low, then it's passive. Doesn't require any work. If you were just to coast down a hill on the bicycle, then that's passive. Doesn't require you to pedal or really do any work. Going uphill, on the other hand, is a lot harder. That takes work. It's going to take some sort of form of energy. So low to high, that's going to be active. So that tells us if it's passive or active. The other thing we need to know is what molecule are we talking about? And that makes a pretty big difference in terms of what kind of passive transport we're talking about. So there's a few different types of passive transport, and they're all really types of diffusion. So first we had simple diffusion. And this was for molecules that were both small and non-polar or uncharged. So oxygen, carbon dioxide, some lipids, things like that. They can move straight through the membrane, no problem. They don't need any special passages. So let's draw a membrane. There's our phospholipid bilayer. In simple diffusion, they move in and out, no problem, just down the concentration gradient. So they're driven completely by where are they most concentrated. They're going to try to spread out to where it's lower concentration. The next one was facilitated diffusion. Facilitated diffusion, it's facilitated by having a passageway, some sort of carrier protein or ion channel. So it needs that because the molecule is bigger and polar, or it's ionic, positive or negative charge. Neither of those things are going to get along with the inside of the membrane, so they need a special passageway through. Which way do they move? It depends on where the concentration is higher or lower. Always from high to low. So when they move, they have to go through the channel, but they're always going to go from high to low concentration. The last type of passive transport is osmosis. And osmosis only applies to water. So if water is moving across a membrane, not just any water movement. So when your heart pumps blood, which is mostly water, that's not osmosis. This is just across a cell membrane. It applies to water, and it's actually a combination of simple and facilitated diffusion. So when water is moving through, a little bit can get through the membrane. And then there are special proteins called aquaporins that allow more of it to move through. Which way does it move? Same as the others, high to low. It moves from where water is more concentrated to where it's less. And that's the confusing part. Remember, it moves toward the area of higher solute concentration. So wherever it's saltier, more sugary, more protein rich, water is going to move there because there's less water over there. So if the cell just made a bunch of water, then it's going to leave the cell. If the cell just took in a bunch of salt, then water is going to follow that salt in. So there's our passive transport mechanisms. Let's talk about active. And there are just two types of that one, primary and secondary active transport. So with this one, let's draw the membrane again as well. And we'll talk about primary and secondary. We can also just abbreviate these with a 1 or a 2 and a little degree symbol afterwards. So you may have seen that. So that's how you can shorthand primary and secondary. So we're going to always move from low to high, and doesn't matter which is in and out in this case, but let's just assume that we're moving molecules in both cases, and that the concentration gradient is low outside and high inside. So it's, in both cases, moving against that concentration gradient. Of course, this is really mostly for ions, some polar molecules as well. So that kind of limits our world. We need to have a membrane transport protein of some sort. So in both cases, we need that. Usually for primary active transport, we call that a pump of some sort. And so it gets its power directly from ATP. So I show it like this. ATP gets broken down, transfers its energy, and work is done moving molecules against their concentration gradient. Then ATP has to be reformed. In secondary active transport, this is a weirder one. So presumably, the cell has already pumped something out, usually sodium. And the most common example of this is going to be in the lining of the intestines. So cells work hard pumping that sodium out. Kidneys do the same thing. And so the concentration of sodium outside is pretty high. So I have a high concentration of sodium outside the cell and low inside. And this is the hard part to remember. That represents stored energy. Sodium wants to come into the cell. And so we're going to let it, but when we do, we're going to use that energy, harness it, and let another molecule like an amino acid or glucose hitch a ride with it. So that yellow arrow there shows that glucose or an amino acid are moving into the cell against the gradient, but sodium is moving with it. So sodium is the power source. So in secondary active transport, we have another molecule moving down its concentration gradient. So one thing's moving with and one thing's moving against its concentration gradient. Whereas with primary, everything is moving against its concentration gradient, but you always have ATP involved. That's how you tell the difference. When it comes to cleaning up our blood, the kidneys basically use all these tricks, passive, active, different points in those tubules that make up all those nephrons in the kidneys. And this is actually really hard work. There's a lot of active transport that goes on to drive this, to clean up our blood so that when we take it back to our heart, it's all cleaned, could go to the cells waste free. Well, there's the urinary system. More complicated than you'd expect, but it gives us a chance to talk about all these really important concepts that will come up again in your future anatomy classes.